Good afternoon, everyone. My name is Hoàng Thị Phương Anh. I am now a PhD student under the supervision of Professor Kim Kyung-woong here in Gwangju Institute of Science and Technology. Today, I'm very honored to be here to share some of my findings in my research about the arsenic accumulation in several crop plants and its mitigation using biofertilizer. You might heard about the sharing of my professor. The arsenic is very toxic element to human health. But do you know that it's class 1 human carcinogen? It can cause many various cancer, child mortality, neurotoxicity, and many lifestyle-related disease. So humans usually exposed to arsenic by the ingestion of food and drinking water with elevated level of arsenic. And the main pathway is by food consumption. So generally, plant uptake arsenic through the root in the environment, but in the case of submerged plant, it can absorb the arsenic through to the leaves by the water phase. So that means that if we are cultivating the crop plants in a high level of arsenic soil, it can accelerate the uptake and accumulation of arsenic in eatable part of the plants, and it will be subsequently presented a potential health risk to the local residents. But unfortunately, there are numbers of studies have shown that the high concentration of arsenic in rice and other crop plants all over the world due to the high level of arsenic in soil and irrigation water, and mostly it's caused by the mining activity. The main reason is that in some part of the world, agricultural practice still occur right next to the mining site without any proper treatment. So given the highly toxic nature of arsenic, it is highly recommended that we should carry out some proper treatment to reduce the transport of arsenic from the environment to the eatable part of the plants. So there are numerous approaches to remediate the arsenic in the environment. But in this study, we focus on an eco-friendly method, that is the use of biofertilizer. So biofertilizer is defined as fertilizer that is consisting of microorganisms and the carrier. Therefore, the biofertilizer can have both effect of the microorganism with specific functions and the carrier. Actually, biofertilizer has been investigated for many purposes, usually to enhance the biomass and the nutrient supplement of the plants. And several studies also reported the use of biofertilizer in reducing the arsenic the heavy metal concentration in eatable part of the crop plants, but little has been done about arsenic. So in this study, the bacteria that I chose is arsenic accumulating bacteria. There are many ways, many mechanisms behind the way that bacteria interacts with arsenic, but it will eventually lead to either reducing or enhancing the bioavailability of arsenic in the environment. So in this study, in order to reduce the accumulation of arsenic in the crop plants, we will use the bacteria that can accumulate the arsenic in their cell, so it would keep the arsenic in their soil. But if we introduce the bacteria alone in the environment, soon after the inoculation, the microbial population will decrease due to unpredictable stress in the environment, such as the change in pH, temperature, salinity, or the presence of other toxicants. So the role of carrier is very important to the survival rate of the bacteria. As you know that lignocellulose material is it has porous structure, so it could provide bacteria with a favorable habitat. So in this study, we chose agricultural waste such as rice straw, rice husks, and coconut husks in order to examined for their ability of absorb arsenic for the carrier. So in order to conduct this study, we follow the outline with the 5 main stages. First, the bacteria will be isolated from arsenic contaminated site, and after that, it will be screened for their ability to absorb and accumulate, to resist and accumulate arsenic. After that, the chosen bacteria will be identified by analyzing the 16A ribosome RNA sequence, and also by biochemical characteristic. Meanwhile, the carrier will be chosen by their ability to absorb arsenic, and after that, the chosen bacteria and the carrier will be incubated together to formulate the biofertilizer. Biofertilizer will be then inoculate into the soil for cultivation, and after that, the arsenic concentration in the eatable part of a plant, also the root part, will be analyzed by RCPMS. So the sampling site for bacterial isolation is Tongsin and Takkum mining in South Korea, because these sites have been reported in previous study with a high level of arsenic in their soil. So you can see that the concentration of arsenic in these soil is quite high, especially from the GJ4 from the Tongsin mining. And after isolation, we collect 32 bacterial strains from these soil samples, and glistening water-like colony was observed in half of the isolate, which is a characteristic of bacterial colony that produce slime and capture. These bacterial structure is the outer layer of bacteria that could help prevent the dewatering or the effect of toxicant in the environment to the bacterial cells. The spore formation was found in several of the bacterial strain. These characteristic of bacteria is the form to resist the extreme condition. So it could be one of the reasons why they can resist to high level of arsenic. So after that, we screen for their ability to resist arsenic by the MIC test. And you can see that overall, the bacterial strain isolated from Dongqing mining exhibit greater ability compared to the Takkum mining, and it's very reasonable because, as I said before, the Dongqing mining site has the higher level of arsenic in their soil compared to the Takkum mining. And the DJ22 and 41 thrive the best in the percent of arsenic. And it's very interesting that these bacteria share the very same characteristic about their morphology. So the strain DG25 and DE12 also exhibit great capacity of their resistance, and as the result of gram staining show that these bacteria has the ability to form their endospore, so that could be one of the reasons why they could resist to high level of arsenic. So all the bacteria that can resist more than 4 millimolar of arsenic will be chosen for the next screening for their ability to accumulate arsenic. So after that, we screen for their ability to accumulate arsenic by incubating these bacteria in the environment that have 2 millimolar of arsenic, and after that we will analyze the arsenic concentration in their biomass, and the results show that the greater arsenic accumulation ability we exhibit in the strain DJ24, and this result is quite similar to the previous study about arsenic. And the interesting thing here is that the strain DJ22 and 41 were among the bacteria that accumulate the lowest amount of arsenic. But in the previous study, we already seen that these bacteria can resist, have the best resistivity toward arsenic. So that means that there are bacteria that have other way to interact with arsenic other than accumulate them in their biocells. But to the purpose of this study, I would choose a bacteria that have great ability to resist and accumulate arsenic. So you can see that the isolate DE12 possess the high arsenic resistance and accumulating ability, but then selected for the identification. The result of the analyzing 60S ribosome RNA show that this bacteria belongs to the family Bacillus, and it show great similarity to the Bacillus megatherium strain. So to make sure that these bacteria can be identified as Bacillus megatherium, we did some biochemical test to assure these results. And you can see that they have very similar results with the previous study about the bacteria that is identified as Bacillus megatherium, and also the characteristic that it described in the Berkeley manual. So the strain DE12 can be identified as Bacillus megatherium based on the analysis of the 16S RNA sequence, and also by biochemical characteristic. The important part is that there is no evidence of Bacillus megatherium about the pathogenic traits, and also the bacteria has been reported to have economic importance because they can produce many commercial enzymes in their growth cycle. So after choosing the bacteria, we did the experiment to screening for the carrier to formulate the biofertilizer. So the absorbent were used in this study was chosen by the absorption capacity using this equation, and this is a result. Because when in order to making biofertilizer, we consider the cost production of the products. So usually to enhance the absorption capacity of the raw material, we usually need to make biochar. But in this study, because this could make the price of the products rise, so we just do the experiment on the raw material, which is rice straw, rice husks, and coconut husks. And you can see that the coconut husks, the result of coconut husks were quite similar to your previous study, which is really low in comparison to rice straw and rice husks. But even though rice straw and rice husks come from the same type of plant, but were a significant difference between them, and these could be explained by the difference in their chemical composition. As you see that the lignin and silica present in rice straw and rice husks with a very abundant amount, but usually lignin is act as a cementing metric between cellulose and hemicellulose compounds, and silica acts as a protecting layer outside of rice plains. So these type of components act as a blocking gate for the accessible functional group to interact with the toxicant. So it's reduced the ability of the rice husks to become a possible absorbent for this study. So we chose rice straw to be the carrier for the biofertilizer formation. The biofertilizer will be formed by incubating the bacteria and the chosen carrier together in 24 hours, and after that we leave it for 1 week in a shade condition, and we will measure the colony forming units inside the biofertilizer. And you can see that it is recommended that the microbial load should be higher than 10 to the 7 cell per gram carrier. In our research, after 7 days of packaging, the formula contained 1.5 10 to 9 CFU per gram, so it is possible to apply into the field. So for plant cultivation, we use the soil in the token mining area, and you can see that these areas, they actually planting rice and crops in this area, so it could be close to the nature of the cultivating condition. So we decided to categorize into 4 groups. The first one is fertile soil, which is non-contaminated. The other one is arsenic-contaminated soil with bacteria, and arsenic-contaminated soil with biofertilizer. And the last one is a control group, which is arsenic-contaminated soil only. So the plants that we used in this research is lettuce, water spinach, and sweet basil, because these type of plant was reported to have the ability to resist high arsenic concentration in the soil, also irrigation water. So I choose this type of plant because their soil from token mining was pretty contaminated with arsenic. So this is the morphology of the plant after cultivating, and you can see that the difference between the length of the plants in cultivated group was not significant, and it was also reported in previous study that there was a poor relationship between the both length and biomass of these plants compared to the one that is cultivated in contaminated soil. So this is the result after we analyzing the arsenic concentration in the shoot and the root of the cultivated plant. And you can see that the arsenic concentration in the root is much higher than in the shoot. It is very obvious because the root is the first part that interact with their toxicants. And the application of biofertilizer actually work because you can see that there is reduction when you apply biofertilizer compared to the control group, but the efficiency of between each group is very different. As you can see that the lettuce and sweet basil actually share the very similar chains because the reduction efficiency is not really high. Especially in case of the lettuce, the application of biofertilizer or bacterial seem to bring very little benefit to the lettuce. But in case of sweet basil, you can see that the reduction efficiency is about 34%. So it's quite high compared to the arsenic concentration in the cultivated soil that we used for this experiment. But in case of water spinach, the application of biofertilizer of bacterial seem to have much better effect than the application of biofertilizer. The difference between lettuce with sweet basil and water spinach can be explained by the difference in the water resin, because when cultivating sweet basil and lettuce, I use the aerobic soil, but in water spinach, we have to use the submerged condition. So it would change the species of arsenic and also the species and the bioavailability of arsenic in the soil. But in this study, the reduction efficiency of when inoculating bacteria alone could reach to more than 50%, which would be a good sign when we apply to water spinach or any other aquatic plants such as rice in the future. So for conclusion, we can say that the strain DE12, which was identified by Bacillus megatherium, exhibit the highest ability to resist and accumulate arsenide, was chosen to form biofertilizer along with the rice straw. And the inoculation of biofertilizer in soil proved to have ability to diminish arsenic content in edible part of lettuce, water spinach, and sweet basil. But in this study, in this presentation, I will only show the results about arsenic, but we do actually analyzing the lead concentration in the edible part of the plant after applying biofertilizer, and it showed very promising results. So we hope that in the future we could conduct another experiment with another plant types, especially rice, about arsenic and also lead in the future. So thank you so much for listening.